About 15 million cases have been reported in the United States of tuberculosis, and 10,000 active cases have been reported in the United States of latent tuberculosis. So it is one of the deadly disease in the later period of your life. Hello everyone, this is Tahir from ZMeds, and today we are going to talk about the causative agent of tuberculosis in detail.
So back to what I was saying. Could be us.
So the cause of tuberculosis is Mycobacterium tuberculosis. In Mycobacterium genera, there are two main species, Mycobacterium leprae and Mycobacterium tuberculosis. So Mycobacterium tuberculosis is the causative agent of tuberculosis. Now, some of the properties of the Mycobacterium tuberculosis are as follows. Mycobacterium tuberculosis has a doubling time really slower than other species. It has the doubling time or the replication time or the growth time of about 18 hours. That is, as compared to the 1 hour, is much greater, and so there will be low doubling of the Mycobacterium tuberculosis. For the growth of Mycobacterium tuberculosis, Livingston-Johnson media is most of the time used. Sputum is used as a sample to test the Mycobacterium tuberculosis, whether test comes positive or negative. If the test comes positive, then it means the patient is suffering from the tuberculosis, and if the test is negative, then it means patient is not suffering from the tuberculosis. How test happens, we will discuss it in later on part of this series about tuberculosis. So in Livingston-Johnson media, egg yolk, and the second thing is malachite green. These two things are used in Livingston-Johnson media, and egg yolk will act as a nutrient, but malachite green is a dye which is used to kill normal flora that is present in the sample sputum. So Mycobacterium tuberculosis is obligate aerobe, that is why it is grown, or it usually grows in the presence of excess of oxygen, and hence infection of the tuberculosis is confined to the upper lobe of the lungs. Next thing is that it is acid fast. So acid fast has a property that they contain mycolic acid. In this case, 78 carbons to 90 carbon atoms are present in the fatty acid structure known as the mycolic acid. There is one important thing, that there is cord-like pattern known as the trehalose dimycolate that is correlated with the pathogenesis of Mycobacterium tuberculosis. Mycobacterium tuberculosis has proteins which reacts or combines with waxes and elicit the delayed hypersensitivity reaction. But same proteins are actually used as an antigen in PPD. What is PPD? PPD is purified protein derivative test. It is also known as tuberculin skin test, and this test is, I will tell you in the later part of the series. Thiocyrrole dimycoserosate is a lipid that must be present in the cell wall of the Mycobacterium tuberculosis in order to cause pathogenesis in the lungs. Mycobacterium tuberculosis is actually resistant to three things: acid, base, and dehydration. Well, by using acids and base such as NaOH, more specifically speaking alkali, okay, Mycobacterium tuberculosis are more specifically resistant to alkali, acids, and dehydration. Now acid and alkalis can also be used to kill other human cells or other normal flora present in the sample, and due to the resistance from the dehydration, Mycobacterium tuberculosis can survive in expectorated sputum that is all dried up. Mycobacterium tuberculosis has become resistant to isoniazid that was only antimycobacterial drug, but now it's finished. And there is another thing, Mdr Multi-Drug Resistance. So Mycobacterium tuberculosis shows resistance to multiple doses of different antibiotics as well. Well, what is the reason? Reason is simple. There is a resistant gene inside the chromosome, not the plasmid, because plasmids are not present in Mycobacterium tuberculosis. So there are two genes, one for the Mdr and other for the isoniazid resistance, which are actually responsible for the resistance of the Mycobacterium towards multiple drugs or antibiotics and isoniazid. And number one is for the catalase peroxidase enzyme that is important for the activation of the isoniazid inside the Mycobacterium tuberculosis, and other one is responsible for the mycolic acid synthesis. So this was all about the Mycobacterium tuberculosis. Next time we will see the rest of the topics, which includes pathogenesis, transmission, treatment, laboratory tests, prevention. So stay tuned.